Paracas. In 1928, beneath the blinding sun of southern Peru, an archaeologist brushed away the desert sand. He expected to uncover the usual remnants of a forgotten coastal culture. Woven textiles, simple ceramics, perhaps typical bones. But the relentless wind revealed something entirely incomprehensible. Staring back from the trench were hundreds of elongated, monstrously shaped skulls. What lay buried in that arid wasteland defied all human anatomy, and decades later, modern science tested the genetic material. The results stopped evolutionary biologists cold. What exactly did those DNA tests reveal? The truth is stranger than history. Here at Echo Mysteries, we are dedicated to unearthing these suppressed archaeological secrets, but we cannot make this journey alone. If this mystery sparked curiosity, please like this video, leave a comment with your thoughts, and subscribe to our channel. Your support isn't just a click, it is the heartbeat of this channel. It gives us motivation to keep digging deeper. Share this with a fellow truth seeker, and let's unravel the forgotten past together. Julio Tello stood on the expanse. It was 1928. The wind howling off the Pacific Ocean carried the chill of the desert below Cerro Colorado. A seemingly unremarkable hill lay a secret that would haunt the scientific world. Tello and his team began to dig. They breached the crust of the earth and found a sprawling subterranean necropolis. Massive, cavernous tombs carved into the rock. Inside, the dead were waiting. Hundreds of bodies were discovered, meticulously arranged in fetal positions, wrapped in complex textiles. But it was not the fabric that stopped the archaeologists in their tracks. It was the skulls protruding from the ancient bundles. They were stretched, they were grotesquely elongated, tapering backward into massive cones of bone. The eye sockets seemed enormous, casting dark, cavernous shadows in the harsh Peruvian light. It was an anatomical nightmare. For almost a century, these Paracas skulls became the ultimate canvas for wild speculation. As the internet age dawned, the fringe theories exploded into a global phenomenon. Amateur researchers and conspiracy theorists declared him the ultimate proof of an extraterrestrial presence. They claimed the cranial capacity was up to 25% larger. They pointed out the apparent absence of a sagittal suture, the connective tissue line that divides a normal human cranium. Videos and blogs circulated unverified claims of independent testing. The rumors were apocalyptic. They said the mitochondrial DNA was unknown. They said it contained mutations that did not fit anywhere on the known evolutionary tree of Homo sapiens, Neanderthals, or Denisovans. They painted a picture of a master race, a forgotten, superior species that once ruled the ancient coast of South America, wiped out by a cataclysmic event. But none of that is the deepest mystery. Between 2014 and 2018, the global scientific community mobilized. Leading evolutionary biologists, anthropologists, and geneticists subjected the Paracas remains to rigorous, peer-reviewed analysis. They extracted pristine genetic material from the ancient bone dust. When the genomic sequencing was complete, the results shattered the extraterrestrial illusions. The DNA was entirely, completely human. It possessed the exact genetic markers of indigenous Native American populations. There were no missing links. There were no alien ancestors. And here is where the timeline of human endurance becomes truly terrifying. If they were human, how could the skull take such a monstrous form? The answer is artificial cranial deformation. It is a practice so physically brutal it challenges our understanding of human survival. Picture the process. Within days of a child's birth, when the cranial bones are still soft and unfused, the head is subjected to relentless mechanical manipulation. Heavy wooden boards are strapped tightly to the front and back of the infant's head. Thick, tightly woven textiles are bound around the skull, creating immense, unyielding pressure. This compression is maintained constantly for months. The growing brain has nowhere to go but up and back. It forces the skull to elongate unnaturally. The supposedly missing sagittal suture was simply fully fused, a biological reaction to the extreme, chronic pressure exerted on the bone plates. The illusion of increased cranial volume was just that, an illusion caused by the radical redistribution of bone mass. Not an actual increase in brain size. This was not the work of visitors from another world. It was a staggering display of cultural engineering. The Paracas civilization was deliberately molding the physical forms of their elite class. They were literally shaping their nobility to look like living deities. The true shock for evolutionary biologists wasn't alien DNA. It was the horrifying realization of how much structural trauma the human infant can endure without dying. It is a testament to the terrifying resilience of our own biology. Just south of the harsh Peruvian sands lies the Atacama Desert in neighboring Chile. It is arguably the driest, most unforgiving non-polar environment on Earth, a desolate wasteland of cracked earth, ancient dried lakes, and jagged salt flats that closely mirrors the dead surface of Mars. In the winter of 2003, amidst the crumbling ruins of an abandoned nitrate mining town called Lenoria, a man searching for historical artifacts unearthed a small leather pouch. He opened it and found a skeleton. It was barely 15 centimeters long. It was roughly the size of a modern ballpoint pen, yet it possessed the hardened, calcified bone structure of a much older living creature. The skull was wildly, impossibly misshapen. It stretched upward into a high, pointed cone. The eye sockets were dramatically slanted and unusually massive for the tiny face. Strangest of all, the skeletal frame contained only 10 pairs of ribs instead of the standard human 12. For an entire decade, this tiny, mummified corpse fueled a massive, highly profitable subculture of extraterrestrial investigators. Elaborate documentaries were filmed, sensational claims were broadcast across the globe. To the true believers, this was the definitive, physical proof of an alien, biological entity that had somehow perished in the barren South American highlands. Was it human, or was it a remnant of something else entirely? The heavily guarded specimen eventually caught the attention of Gary Nolan. He was a highly respected immunologist and geneticist working at Stanford University. Unlike many of his academic peers, who immediately dismissed the artifact as an elaborate hoax or a deformed monkey, Nolan approached the anomaly with a quiet, icy objectivity. He agreed to examine the controversial remains, accepting the specimen directly from the hands of passionate UFO enthusiasts. He brought the tiny skeleton into one of the most advanced, sterile genetic laboratories on the planet. Nolan held no preconceived judgments. He did not care about the media circus. He simply wanted to read the biological code locked inside the ancient bones. His specialized research team meticulously extracted DNA directly from the bone marrow of the impossibly small ribs. They painstakingly prepared the genetic samples and fed the data into state-of-the-art sequencing machines. They were hunting for the fundamental blueprint of the creature. The supercomputers hummed relentlessly for days, mapping out billions of base pairs of genetic information. When the complete whole genome sequencing was finally rendered on their digital screens, the entire extraterrestrial hypothesis vaporized in an instant. Every sequence, every complex marker, every single strand of DNA aligned perfectly. It was a direct, undeniable match with the human reference genome. There was no missing link. There was no unearthly origin. The DNA belonged definitively to a human female of indigenous South American descent. The Atacama alien was actually a human girl from the local desert region. But here is where the genetic analysis becomes particularly devastating, because simply being a human being did not explain the catastrophic physical deformities of the skeleton. On March 22, 2018, the official, peer-reviewed findings were published in the prestigious journal Genome Research. The paper detailed a level of biological mutation that stunned the global scientific community. The Stanford and UCSF researchers discovered 64 distinct, exceptionally rare genetic mutations scattered aggressively across 7 different genes. These were the specific genes intrinsically linked to skeletal development and cartilage formation. The girl had suffered from a violent, unprecedented combination of severe dwarfism, extreme facial malformations, and a terrifyingly rapid premature bone aging disorder. Her skeletal structure had aged so rapidly inside the womb that her bones practically resembled those of a 6-year-old child, even though the biological evidence proved she was merely a prematurely born fetus. The fundamental building blocks of biology had simply turned against her with cataclysmic precision. The Atacama alien was not an advanced invader from a distant, unknown galaxy. She was a deeply tragic anomaly, wrapped carefully in cloth and buried inside a leather pouch near an abandoned desert church. She was clearly mourned. They likely watched her die shortly after, or perhaps even before, she took her very first breath in that harsh air. The real terror for evolutionary biologists was not the fictional prospect of alien life. It was witnessing DNA coding errors leave the desolate sands of the Atacama Desert. In 2020, researchers at UCLA embarked on a different kind of excavation. There were no shovels involved in this expedition. There were no dusty tombs, no mummified corpses, and no bones. Instead, they descended directly into the digital architecture of human existence, utilizing massive supercomputers to sift through genomic data. They sifted through the data of hundreds of modern individuals from various West African populations, hunting for evolutionary markers. The scientists expected to find the familiar, well-documented traces of our ancient ancestors and the standard biological timeline. The complex mathematical algorithms were designed to simply map the known branches of the human family tree through generations. But the machines found something that fundamentally did not belong in the code as they processed astronomical genetic information. The predictive algorithms began isolating strange, unexplainable bands of DNA, thick and completely foreign sequences embedded deep within. It was like finding a chapter from an unknown language hidden inside a familiar history book. Scientists cross-referenced sequences. They cross-referenced these mysterious sequences against the fully mapped DNA of Neanderthals. But it was not a match. They ran the data against the genetic profile of Denisovans, another known archaic human species. But it was a failure. The code did not align with any known human ancestor. It did not align with any primate in the global biological database. It was an anomaly of incomprehensible proportions. It was a massive, undeniable phantom signal echoing from a dark, prehistoric void. And here is where the digital excavation becomes truly chilling. On February 12, 2020, the UCLA researchers published their terrifying conclusion. The anomalous code was not a computational glitch or a corrupted file. It was a ghost. The rigorous statistical analysis revealed that anywhere from 2 to 19% of the genetic heritage in these modern West African populations originated from a completely undiscovered species. This ghost species is believed to have violently split from the central human family tree hundreds of thousands of years ago. They were an entirely parallel humanity, a shadow race that evolved, hunted, built communities, and thrived in the ancient world. Yet they were completely erased from the physical fossil record. We have absolutely no bones to study, no elongated skulls. We have no stone tools or crumbling archaeological ruins to prove they ever existed on this planet. Yet, the biological proof is irrefutable. Written permanently into the blood of living humans. Sometime in the deep, unrecorded history of prehistoric Africa, ancestors encountered this phantom species. They did not just compete for limited resources. They interacted, and they interbred. The resulting hybrid offspring absorbed the alien genetic material, carrying it forward as an indestructible biological archive through the collapse of empires and the shifting of continents. This realization disrupted everything. The human story was suddenly no longer a clean, straight line of progressive, isolated evolution. It was revealed as a tangled web. The most unsettling aspect is not that this shadow species disappeared. The terrifying truth is that they never actually went extinct. They are walking among us today, quietly living inside our DNA. Siberia. Deep within the Altai Mountains lies Denisova Cave. It is a massive, freezing limestone cavern that has served as a brutal shelter for various human species for hundreds of thousands of years. The conditions inside are perpetually cold, dark, and perfectly suited for preserving biological material. Over the decades, archaeologists have meticulously sifted through layers of ancient dirt, extracting countless thousands of fragmented bones. Most of these fragments belong to long-extinct animals, woolly mammoths, cave hyenas, and ancient horses. Identifying human remains in this chaotic graveyard of pulverized bone is an agonizing, microscopic process. In 2012, Russian researchers unearthed a completely unremarkable piece of bone. It was barely 2 centimeters long. It looked like a piece of debris. It was so small and insignificant that it was simply catalogued, bagged, and sent to the Max Planck Institute for Evolutionary Anthropology in Germany for routine collagen analysis. They assumed it belonged to an animal. They had no idea they were holding a biological impossibility. The researchers at Max Planck subjected the tiny fragment to a rigorous screening process. They used specialized lasers and advanced mass spectrometry to identify the specific protein signatures hidden within the marrow. When the results flashed on the screen, they confirmed the bone was indeed from a hominin. It belonged to a young girl, roughly 13 years old, who died inside the freezing cave approximately 90,000 years ago. The team affectionately named her Denny, but to truly understand who she was, they needed to drill into the bone and extract her nuclear DNA. They needed to map her entire genetic inheritance from both her mother and her father. When the sequencing machine finally completed its complex rendering of Denny's genome, the scientists stared at the data in absolute disbelief. The results were so fundamentally impossible that the lead researcher immediately assumed the highly sensitive equipment had malfunctioned. They suspected massive laboratory contamination. They believed someone had accidentally mixed the genetic samples of 2 completely different archaic human species into a single vial. But there was no error. The machines were functioning perfectly. The data was terrifyingly precise. Denny's mitochondrial DNA, the genetic code inherited exclusively from her mother, identified her mother as a pure Neanderthal. But when they analyzed the rest of her genome, the code inherited from her father, it identified him as a pure Denisovan. On August 22, 2018, the Max Planck Institute published their apocalyptic findings in the journal Nature. Denny was not just a member of an archaic human species. She was a first-generation hybrid. She was the direct, immediate offspring of 2 completely distinct human species that evolutionary biology had always categorized as separate. And here is where the timeline of human evolution completely shatters. Imagine the astronomical mathematical odds of this discovery. For centuries, the rigid scientific definition of a species dictated that distinct biological groups do not routinely interbreed. Neanderthals and Denisovans had separated on the evolutionary tree hundreds of thousands of years before Denny was ever born. They evolved in completely different environments, developing distinct physical traits and genetic markers in isolation. Yet, somehow, in the freezing shadows of a Siberian cave, these 2 distinct branches of humanity collided. But the true shock for evolutionary biologists wasn't just that they interbred. It was the statistical impossibility of finding Denny. Out of the incredibly scarce, minuscule number of archaic hominin fossils ever discovered and sequenced in human history, Scientists managed to stumble directly upon a first-generation hybrid. The odds of finding a pure F1 hybrid are practically zero. Unless, of course, this kind of interspecies mixing wasn't rare at all. What if the ancient world was a melting pot? Denny's tiny bone single-handedly vaporized the concept of pure human lineages. It proved that different human groups merged. They shared fires, they shared shelters, and they shared their DNA. Return to the high coastal deserts of Peru. High above where the Paracas skulls were buried, the Andes Mountains tear through the clouds in a hostile environment. At elevations exceeding 4,000 meters, the air is terrifyingly thin. Oxygen levels plummet to levels that cause hypoxia. This would cause pulmonary edema and eventual death for the vast majority of humans currently living on the Earth. The temperatures are brutal, the ultraviolet radiation is intense, and the available food sources are drastically limited. Yet, for thousands of years, indigenous Andean populations have not just survived in this vertical wasteland, they have thrived. For decades, scientists understood that modern high-altitude populations possessed unique physiological adaptations to handle the extreme lack of oxygen. But they assumed these changes were the result of a slow, gradual evolutionary process stretching back over tens of thousands of years. They believed human DNA required vast amounts of time to permanently rewrite its own survival code. In 2018, geneticists decided to test this assumption. They journeyed into the freezing Andean peaks and extracted DNA from the skeletal remains of ancient Peruvians who lived in the region roughly 7,000 years ago. They wanted to see the baseline. They wanted to observe the slow, methodical pace of human evolution in real time. But when they mapped the ancient genomes and compared them to modern Andean populations, the timeline completely collapsed. The genetic data revealed a speed of biological adaptation that was previously considered scientifically impossible. On November 8, 2018, the findings were published in Science Advances, detailing a genetic explosion. The researchers discovered that the crucial genetic mutations responsible for high-altitude oxygen processing did not evolve slowly. They swept through the entire population with terrifying, unprecedented velocity, and here is where the story of human endurance becomes truly unsettling. The DNA analysis proved that it wasn't just a single gene that mutated. It was a massive, coordinated genetic overhaul, within an incredibly short evolutionary window of just a few thousand years. The ancient Andeans rapidly developed a complex suite of adaptations. Their DNA fundamentally altered how their bodies processed starch. To maximize energy from a limited diet, it radically enhanced their immune systems to fight off specific, deadly pathogens unique to the mountain environment. Most astonishingly, their genetic code permanently restructured their cardiovascular systems to efficiently pump oxygen-starved blood without causing fatal heart failure. This wasn't a slow biological drift. This was forced, hyper-accelerated evolution driven by extreme environmental stress. The human body was essentially weaponizing its own genetic code to conquer an uninhabitable landscape. The realization sent shockwaves through the field of evolutionary biology. It proved that Homo sapiens are not just passive passengers on the slow train of evolution. When pushed to the absolute limits of physical survival, our DNA has the terrifying capacity to mutate, adapt, and rewrite its fundamental programming at speeds that defy our conventional understanding of biology. The ancestors of modern Peruvians did not wait for evolution to slowly catch up. They biologically conquered the Andes in record time. It forces us to ask a terrifying question. If the human genome can radically redesign itself to survive the crushing lack of oxygen in the high Andes in just a few millennia, what other dormant, extreme mutations are currently sleeping in our blood, just waiting for the next cataclysm to wake them up? From the elongated skulls of Paracas to the tragic mutations of the Atacama Desert, from the phantom DNA of West Africa to the frozen hybrid bones of Siberia, and finally, back to the hyper-accelerated evolution on the peaks of the Peruvian Andes. The physical evidence is undeniable. The rigid, comforting timeline of human biology we were taught in schools is completely fracturing. We are not a pure, isolated species that simply walked a straight path out of antiquity. We are a chaotic, deeply resilient mosaic of interspecies breeding, radical adaptation, and terrifying genetic mutation. The real mystery is what our DNA is capable of becoming.